Hello and welcome to O-Levels Biology lesson. Today we are going to focus paper 1, the topic Biotechnology and Genetic Engineering. Let's explore. Okay. Our question 1 indicates that a gene of insulin is taken from human and it is placed in the bacterium. The bacterium can make human insulin. So let's understand diagrammatically. This is the diagram of human cell, and this is the diagram of a bacterium cell. So if we consider the structure of bacterium, bacterium has the linear DNA and a plasmid ring. Now, what do we do in the technique of genetic engineering? We take a small segment of human DNA and we potentially transfer it into the plasmid ring like this. And after this entire process, the bacteria become modified and it will start to produce insulin. So they're asking that what is the best term to represent this process? It is actually genetic engineering. In second question, they are asking us graph. It's actually a graph related question. We need to comprehend the situation. Okay. The penicillin is produced in a fermenter. So we need to understand what are fermenters. These are actually huge containers. Sometimes they are used to make any food product like yogurt, cheese. Sometimes they are used to make different type of proteins like antibiotics. So here they are explaining us that in a fermenter, the penicillin is produced. Two terms are a bit confusing. We need to get a grip on it. Penicillin is antibiotic, and penicillium fungus is used. It is actually a microorganism used. The graph shows the mass of penicillin, the mass of living penicillium, and the concentration of penicillin changed over the time. So it's a graph that is representing a 5-day data. With the passage of time, we need to observe that as the time passes, the mass of penicillium was increased, and after some time, the mass of penicillium started to decrease. But on contrary, when the penicillium reached to its maximum, the production of penicillin started here, right? And it reached at its peak on day 5. So now what does the question ask? The question is related to the the production. Which is the best time to collect penicillin? So is it day 1? Is it day 2? Is it day 3, 4, or 5? So if we analyze the graph, we come to know that on 5th day, the concentration of penicillin is maximum. So according to this graph, which is the best day, 5th day is the most ideal condition where I can acquire maximum amount of penicillin. Question number 3, when human DNA is inserted into the plasmid ring, as I mentioned in question number 1, so which enzyme is used to cut the DNA? To remember it, I have made a mnemonics of cut, of scissor and tape. Okay. If we want to cut the DNA, we need scissor, an enzyme that work like a scissor. And if we want to paste the DNA, we want an enzyme that work like a tape, right? So the enzyme DNA ligase, DNA DNA ligase act like the tape. It help in pasting. And restriction enzyme is actually used to cut the DNA. So if we want to cut the DNA, which enzyme we will be using? The restriction enzyme. Okay. Now, question number 4. Here are the different stages mentioned in the sequence, and they are asking which of the stage involved in the formation of the sticky end. So first, we need to understand what is the sticky end. This is the human DNA, and this is the bacteria plasmid ring. Okay. During the like this entire process of genetic engineering, first we need to make a cut in the human DNA, and we also need to make a cut in the plasmid ring. Okay, these cuts should be appropriate in a way. We are not going into the detailed biochemistry, but the ends should be cut in a way that they tend to make the sticky end. Why it is necessary to make a sticky end? So the next time when we insert the plasmid ring, they are attached to each other. So actually step number 1 and step number 2. The DNA making up human gene is isolated with the help of restriction enzyme. When we cut, we make sure the sticky ends are made. And the DNA of the plasmid ring is cut open with the restriction enzyme. This must be ensured that these sticky ends are made. Which term is used for the transference of gene? It's the genetic engineering term is used to transfer the DNA from one organism to another. What is the role of anaerobic respiration uh, in making bread? Oh, we missed question 5. In question number 5, they are asking why is yeast used in making bread? Yeast is a microorganism and it respires anaerobically. And when it respires anaerobically, it makes two of the products. One is ethanol and the other is carbon dioxide. Right? So ethanol is a kind of alcohol. For now, just leave it. If we concentrate on carbon dioxide is a gas. For example, if I am making the dough, initially the dough is flat. But uh, uh, if we leave this dough over a period of time with yeast, we notice that it start to rise. So which of the chemical substance is actually making the dough rise? It is actually the carbon dioxide. The carbon dioxide is made uh, during the respiration, which make the dough rise. Now, question number 7. What is the role of anaerobic respiration in making bread? We did just earlier. Here, like when we are making dough, we actually need carbon dioxide gas that make the dough rise. Question number 8 is bit interesting, and it is actually considering the use of considering the production of two things, and beer and bread. Okay, we use yeast to make both the products, and we know that when yeast respire anaerobically, it is making two of the substances. One is ethanol, and the other is carbon dioxide. Ethanol is ingredient of wine or alcohol. So when if I am an industrialist and I am making beer, so I am actually interested in ethanol production. If I am an industrialist and I'm running my bakery, so I am interested in the production of carbon dioxide. Okay, why is yeast used in making bread? The anaerobic respiration produces carbon dioxide. Like for example, if I made the dough and I cover it with some fabric or cloth and I put it in a cupboard that is closed. So actually, I am preventing the fresh air to come inside, and I'm trying to make the medium anaerobic. So the yeast do not get oxygen and the yeast will start to respire anaerobically. When it will respire anaerobically, it will produce carbon dioxide. Question number 10. Okay. They are asking, okay, in question number 10, when making commercial apple juice, like for example, you have some fruit and you smashed it in a blender. So once we do it at home, we notice that the juice is turbid. It is unclear. What is the reason behind it? Actually, within this extract, there are some the pulp of fruit, and this the cells in the pulp, they are connected together. Let's read the question. When making commercial apple juice, the fruit is crushed and release the cell content. Chemical Q is found between the cells, like that what I was talking about. It holds the cell together, and due to the presence of these groups of cells, the solution is turbid. If we use an enzyme, so this enzyme actually breaks the bond between these cells, right? And each cell gets separate. It has two effects. Number one, the quantity of juice increase and the solution become clear, the juice become clear, right? So they're asking that which material it is. If we this question is related to the topic cell structure and organization. The two cells are connected together with the help of material pectin, cellulose, hemicellulose. So if we want to break the pectin substance, so which enzyme I should be using? I would be using the enzyme pectinase that will digest the wall of the chemical Q. Like which chemical was substance was holding the cell? It was pectin. When pectinase acts on it, individual cell gets separated and the solution becomes clear. So this is the correct answer.